Hi everyone, I'm Jaya Vishwanathan, and you've found the No Brainer podcast where I discuss all things neuroscience in delightful conversations with various experts. I hope you enjoy today's episode. Hello, everyone, and welcome to another episode of the No Brainer podcast. And I'm so excited to invite our guest today, Dr. Malú Gámez Tansey. She's a mentor of mine as well. And Dr. Tansey is the Norman and Susan Fixel Chair in Neuroscience and Neurology, as well as the former director of the Center for Translational Research in Neurodegenerative Disease at the University of Florida. Her lab focuses on the role of inflammation and immune system responses in brain health, as well as neurodegenerative disease, with particular focus on the central peripheral neuroimmune crosstalk and the gut-brain axis. And don't worry about any of those. We're going to dig deeper into all of these topics. Her long-term goal is really to develop better therapies to prevent or delay these various neurodegenerative disorders. Dr. Tansey obtained her BS and MS in biological sciences from Stanford University and her PhD in cell regulation from the University of Texas Southwestern, followed by postdoctoral work in neuroscience at Washington University. And then as head of the chemical genetics at Xencor, she co-invented novel soluble TNF inhibitors that have now advanced to clinical trials in Alzheimer's disease. She returned to academia as an assistant professor of physiology at UT Southwestern, and then was recruited to Emory University School of Medicine as a tenured associate professor. And then after 10 years at Emory and rising to the rank of full professor, where she has earned several mentoring awards from students and faculty for her efforts in championing early-stage investigators, women, and other underrepresented groups in STEM. She was recruited to the Department of Neuroscience in the College of Medicine at the University of Florida, where she served on the executive committees for the McKnight Brain Institute and the Fixel Institute for Neurological Diseases. She will be moving now to the Stark Neuroscience Research Institute at Indiana University in Indianapolis in January of 2025 as the first director of neuroimmunology research and the executive associate director of education at the Stark Institute. So with that mouthful introduction, you can see why I'm so excited to have the opportunity to talk with Malú about all the exciting research that's happening in this space that Malú is championing and cheerleading, and also all the many ways in which she's supporting the careers of neuroscientists who are coming up the career ladder, and has just the most valuable advice that I've received personally as well. So with that, I'm gonna get started here, Malú. So I have so many questions for you, as you know, and our conversations always span everything. But let's start with, for me, you've become the leader in neuroimmunology, which is, the immune system and the confluence with the central nervous system or peripheral nervous system, and neurodegeneration before people really knew to put these two things together. So can you tell us a little bit about your journey and what you enjoy about your work, as well as some of the research and clinical questions that you work on?
Sure. First of all, thank you so much for having me. This is so exciting for me. You are incredibly kind with your introduction, and I am one of many people who is trying to raise awareness and maybe stir the pot a little bit with regards to thinking outside the brain. Right? That's how we are thinking outside the box, by thinking outside the brain and the influences that other organs, in particularly the gut and the peripheral circulation, have on brain health and brain function. And how it all started was way back when I was a postdoc. I learned a lot about neurotrophic factors GDNF, nurturin. I was very fortunate to be in Gene Johnson's lab at WashU as a postdoc, where some of these ligands were discovered that were very potent neurotrophic factors for dopaminergic neurons. And so I learned a lot about how to think about cell death. And it was clear then that the cell death that people were looking at for neurodegenerative diseases was not necessarily because they were missing trophic factors. It seemed like there might be some toxic element or some deficit that developed maybe with aging and would give you risk for Parkinson's and Alzheimer's. And so Parkinson's being, where you lose your dopaminergic neurons. In Alzheimer's, you lose basal forebrain cholinergic neurons. And so it affects different behaviors, right? One is the motor, the other one's the cognitive. But there's a lot of overlap in these features, and there are some commonalities to these diseases. And when I left my postdoc, I went to industry, and I thought, I'm going to industry, I'm going to make a lot more money, work fewer hours. But in truth, I always say that once a workaholic, always a workaholic. And so even if you have an 8 to 5 job, you still take it home and you're still thinking about it, which is fine, as long as you try for the work-life balance and work-life integration. So it was in biotech that I was handed a project having to do with tumor necrosis factor, or TNF, which is one of the more important inflammatory drivers of, chronic inflammation. But it also is important to fight infection. So you need it for immunity. And it's also really important for myelination of your nerves. So I had to work on this TNF project. I became really interested in inflammation. And it's kind of funny because when I was a graduate student, I had lots of friends that were immunologists. And I thought that was like the worst possible, discipline. And my friend Mary Kirianopoulos said to me, oh, get over yourself. A cytokine is the same thing as a growth factor. It just has a different name. And she was right. And so I ended up really falling in love with the inflammation field and appreciated how complex it was and how many things it's important for. And I started reading about what about inflammation in the brain, right? And so at the time, it was really thought that the brain was immune privileged and that there was no real immune interaction and no immune cells from the periphery going into the brain, that they had their own brain resident cells like the microglia, and that was it. And through the decades, 2 decades now, we've come to appreciate that that's not really the case. The blood-brain barrier is a biochemical barrier, not a physical barrier. And so the brain is immune specialized, and you have cells coming and going with very specific purposes. But it is true that with aging and certain conditions, in particular chronic systemic inflammation, you create conditions that make it leakier, right? And so that leakiness dysregulates the traffic across that. And so when I started looking at what is the role of inflammation in neurodegeneration, because that's what I had known as a postdoc, I thought of putting the two topics together. And when I went back to academia after 2 years in biotech, that's what I started my lab on, the role of inflammation in Parkinson's disease and disease models. And that was 2002. There was no interest, no what are you talking about? That's not a thing. And so it was tough, but I thought that we had the opportunity to at least test some hypotheses, and we had the tools to do it because in biotech we developed these soluble TNF inhibitors, which were not available at the time. All the tools to interrogate TNF function were basically not able to distinguish the soluble form of the cytokine and the membrane attached form of the cytokine. And they have very different biologies. And so what happens is when you block both forms of the ligand, you end up with some side effects of some of these medications. So these are the Enbrel, the Remicade, the Humira. They're antibodies that are blocking both ligands, membrane bound and soluble. And the Enbrel is a decoy receptor that acts as a receptor and sops up both the soluble and the membrane bound. And so what happened is these were amazingly important drugs to control Crohn's and psoriasis and inflammatory bowel disease and rheumatoid arthritis. But it had a black box warning saying, if you have an active infection, don't take this medication. And so what that did is it opened up the possibility that maybe we had to develop better TNF inhibitors that didn't also cause the opportunistic infections and the demyelinating symptoms, that we needed to just target the soluble part of the pathway. And so that's what ended up happening. And these tools that we created and co-invented in biotech, I took them to my lab at UT Southwestern through a material transfer agreement. And I basically used them to find out which pathways were soluble TNF dependent. Is the dying of neurons in a Parkinson's model soluble TNF dependent? And so we would put them in the brain, we would put them in the cells and culture. And that's how it all started. And 20 years later, I'm really proud to say that, they are in phase 2 of clinical trials for MCI and Alzheimer's, based on a lot of publications from 70 plus labs, not just mine, showing that soluble TNF is probably a really important actor in dysfunction of synapses, cognitive decline, in oxidative stress. And we think that by targeting it, we may be able to delay, arrest, maybe slow down the progression of some of these age-related diseases where you have this chronic inflammation in the brain, and where we think the chronic inflammation in the periphery and in your gut is also able to affect your brain because of what I said, that it's not immune privileged. So that's the paradigm that we're focusing on, the role of systemic inflammation, the role of gut inflammation, on how it affects your brain through the ages. How does a good diet change that? How does exercise change that? The gut microbiome, will it give us any clues about how these diseases start and develop? And is there a way to intervene at that point where, for instance, in Parkinson's, when you have GI dysfunction, it tends to happen like 1 or 2 decades before you get your motor, symptoms of tremor. And in a neurologist's office, that's what they mostly use to diagnose your Parkinson's. And that's already pretty advanced. So we'd like to think about a way to diagnose earlier, to look at what we call the premotor or prodromal stage of Parkinson's, where there's GI dysfunction, anxiety, sleep disturbances, and maybe find the individuals that are inflamed and have those early features, and then put them in a clinical trial to try to arrest that conversion, say, to motor Parkinson's.
Wow. So you've covered so many topics there, and I want to pull at a few different threads. Sure. But what I was really intrigued by is this notion of maybe using these kind of chemical messengers going from the periphery to the central, to the brain, which is this organ that sits in this special area and is protected by the blood-brain barrier from anything just crossing into it willy-nilly. And then, so kind of finding pathways there to kind of interrupt this endogenous talk between what's happening in the periphery, the rest of your body. And what I really find amazing about your work is also that the fact that it's so technical and specific, but at the same time, it's far-ranging, and it covers, as you said, things like diet, like motor function and cognitive function and behavioral dysfunction. And so I know that you've published like close to like 18, 20 articles just in the last year. And so maybe can you tell us a little bit about like how you're picking these questions to focus on that are so technical? Because when I look at it from the outside, to me, the options are a little bit mind-boggling. And I'm not, as you know, I'm not like in the immunology field at all. But so you mentioned that, yes, TNF alpha or TNF, which is tumor necrosis factor, is pretty important. But then, like, how did you figure that out of all of the different trophic factors? And how does that science work on like a more conceptual level?
Yeah, so I mean, that's a great question. I stumbled upon TNF because I was in biotech and it was a TNF project. It was a project
I see.
to make better TNF inhibitors, right? And I was
Yeah.
like, oh, good God, really cytokines? But I was the only biologist and I did
I
it,
see.
right? And so it was a kind of project where it was real team science, where
Yeah.
you had let's make a TNF variant, right? A mutated TNF that will have no activity as a TNF, but will inhibit the real TNF. And someone very clever decided that, the TNF superfamily is generally trimers that act on 3 receptors. So it's a 3 on 3-fold symmetry. It's actually very beautiful. And what you have then is you have monomers of, say, TNF, right? 3 of them contacting each other. And then each one contacts 2 receptors, right? So it's a beautiful 3 on 3-fold symmetry. And what someone decided is, let's put a mutation right at the receptor ligand interface, single point mutation, 1 on each side, and that will disrupt binding. And if you do that, it looks like a normal TNF, but it has 2 point mutations that when you give it to receptor, they're not binding because they've got very limited binding affinity. But what happens, maybe by some stroke of luck, they don't all work well. We were hoping they did, but they really don't. What happens when you have the trimer, right, of the normal TNF, it comes off. There's an off rate where the that can now come in and heterotrimerize with the native TNF. So you end up with these heterotrimers of mutant TNF, good TNF, or 2 mutant TNFs and 1 native TNF. So what this TNF mutant does is it sops up the soluble species. Now, the reason it doesn't affect the membrane bound is because the membrane bound is a trimer, but it's stuck on the membrane. There is no off rate on the trimers. So the mutant TNF can't get in there. So it makes it a really beautiful and specific inhibitor of the soluble TNF.
Right.
So it was kind of serendipitous that the first family member that we happened to pick and go through put the mutations, the molecular biology, the in silico group put in the mutations in a way that they didn't disrupt binding. The molecular biology group made the libraries. The protein chemistry group made the proteins. And my group, which was the chemical genetics group or cell biology group, was at the end of the assembly line where we just designed assays to see if these proteins really had new and improved properties, right? And so the company had been doing a lot of mutations, but mostly they had industrial contracts like DuPont and Dow and, making changing the pH optimum of some enzyme that was used in some industrial process. This was the first biology project. And so it got me thinking about TNF and inflammation. And then I went back to academia. I'm sure you can ask me why. I loved being in biotech. It was, it was like sprinting, this and then this and then this, keeps you on your toes. But I did find it a little bit unstable in terms of, the business plan can change the trajectory of scientific careers. And I thought, wow, I'm not sure that I like that. It was LA. And so, we had a mortgage, we had children. And I said to my husband, I said, I think we need to reconsider academia, right? And so I, applied for a job that had been offered to me before I went to biotech at UT Southwestern. And I said, hey, is that job still open? And they said, well, it depends on what you've been doing. And it turns out
Yeah.
that this was a really good story that was being
Yeah.
considered for publication in Science. And
Mm.
when that happened, that certainly, I know, helped me make the jump from biotech back to academia, right?
Right.
That I had some deliverable, something that I had done for those 2 years that
Yeah.
moved the science forward. And so that was the foundation of my interest. And the way we pick questions now, this is going to surprise you.
Okay.
It's based on who wants to do what when they come to my lab. So
Okay. Okay.
we have a general umbrella of we're interested in immune and inflammatory mechanisms that affect
Okay.
brain function and that maybe lead to neurological disease, but we're not married to any particular disease, right? We started in
Okay.
Parkinson's, we've done some Alzheimer's, we've done some FTD, we've done some ALS and depression and even autism. And so it's really driven by the interests of the people who come to my
Mm.
lab and say, what about the gut, right? I had Madeline
Mm.
Hauser, who was an immunology student, said, what about the gut? And I said, what about the gut? And so they've taught me everything that I know. Um,
Yeah.
and we try to go in directions where we think we can make a contribution, either because of our where we've been in terms of our knowledge or where we have tools or can develop tools, right? We're
Right.
not necessarily going for the hottest thing or the most protected thing that nobody works
Mm.
on. I really have committed my career to developing the next generation. So if a postdoc
Mm.
comes in and says, hey, I'm interested in depression, I said, okay,
Yeah.
let's work on developing a depression model and maybe don't reinvent the wheel.
Yeah.
Go find one that works or collaborate. And so that's how it's been. And so it's really driven by the team, by the people who come to my
Right.
lab, and they they work with each other, they synergize, they help each other. A lot of our work involves mice, and a big takedown of a big cohort. You can't do it with one person because you're taking blood and brain and gut and all these tissues, and you have to process them right away. So it's an assembly
Mhm.
line, and it's real team science. And I think they learn
Mm.
to help each other. And today I do it for you, and next time you do it for me. And they learn project management because they have to plan ahead their ballet of, well, in mid-September I'm going to sock 80 mice. I need X number of hands on deck. So
Mm.
it's a good
Mm.
experience as well.
Absolutely. And I actually love that approach. I think that's what makes you such a great mentor, that people are constantly wanting and lining up to really work with you, is
Aw.
because you are so open to hearing the ideas of what other people want to work on and maybe have from their experience and background. As we know, there are a lot of labs that don't work that way as well. And
Right.
so it's a breath of fresh air, because really the success of your research, I'm not even talking in terms of publications, because I know that is the currency in academia, but just in terms of like the skill sets of the people coming out of your training programs and your lab, and the diversity of the topics that you're investigating and providing insight into, I think that's a real testament to your approach of actually being a mentor with an open door policy, where it's a two-way relationship. And I
Yeah.
actually absolutely love that you're doing that in your lab. That's
Yeah,
fabulous. Um,
we oftentimes emulate what we learned and how we grew up in our
yeah.
own scientific homes. My mentor, Gene Johnson, used to say, if you're not having fun, you're not doing it right. And don't worry about the
Hmm.
competition. Don't look over your shoulder. Do the best experiment you can. Do the killer
Yeah.
experiment, right? The experiment that's going to blow the hypothesis out of the water. And he would also say something
Right.
that really resonates, which is, if you only had so much time and so much money, what would be the killer experiment that you would want to do? Like, don't,
Right.
dot the I's and cross the T's. Just what is the one experiment that's going to say, I'm onto something, or this is not,
Not.
a good proposition. But I
Right.
think the thing that you said is really true. we're just training people and giving them a set of tools, right? How to think, how to ask good questions, how to troubleshoot, how to scrape yourself off the sidewalk when something doesn't work, how to deal with rejection of papers and grants, how to build, the network of people that are going to be your colleagues, and how to, balance the writing the papers, the writing the grants, the reviewing, the teaching. It's a lot. I mean, you have to be a good multitasker, I think, to make it in this field.
Absolutely.
But most important, you have to work on what you really love, right? If you put someone on a project that they hate, they're not going to produce good work. That's just a non-starter for me. So, I have told people, no, we're not going to do that. But it's not because I have a preconceived notion of what we should work on. It's usually like, well, we don't have the tools. It'll take too long. It's not my expertise. I really can't mentor you in that too much. Like a student, they can't take on risky stuff. They're supposed to get in, get out, learn how to ask questions, and move on to bigger and better things. But I have had a lot of people who have not gone into academia. I've had people who have gone into science policy and foundation work.
Right.
I have people in biotech, and I have people who went to academia and, got tenured. It's just very specific to whatever you want to do. So I don't have the bias. You want to help develop their career and where they want to go.
Yeah. That's just beautiful, Malú. May I just say that? I
can't
I'm a
imagine
fan.
any other way. I can't imagine any other way to do it, honestly. It would not be right. Yeah.
I want to take a step back into the science of it all, and there are a couple of questions that I would like to maybe pick your brain on. So one is, I think the blood-brain barrier that you mentioned in the first part is quite a fascinating thing that many people, including people in the audience, may not quite understand. And I think you touched upon a few things, like it's not a physical barrier, it's a biochemical barrier. And if you can expand on that a little bit, and then get back to this idea of what makes brain immune cells different from, say, immune cells that are in the rest of the body, or are they the same, or what's really been difficult about targeting the immune system specifically in the brain, given the fact that the immune system, as we now know through your work and others working in this area, that it's now called the systems biology of inflammation, because it contributes so much to all of these diseases in different ways, also based on different risk factors. And so as we are trying to work towards, and I know you are as well, this idea of precision medicine, where we want to treat people for what they have. So just a little bit of idea on what is the difference between immune in the brain, the rest of the body, and what is this blood-brain barrier, and why is it been so difficult to target the brain in treating these diseases?
Those are really great questions. So I think a couple of things in terms of the blood-brain barrier. One thing you need to recognize, and I think this is true for all organisms, is that every place where you're in contact with the environment, there's going to be a barrier. Okay, there's a barrier in your skin, so there's a skin microbiome. There's a barrier in your nasal epithelium. So there's a nasal microbiome. There's vaginal microbiome, lung, and of course the gut, right? And so these barrier sites, including the blood-brain barrier, are not physically walled off structures at all. They contain, in terms of the blood-brain barrier, they contain astrocytes, they contain pericytes, they contain endothelial cells, and the endothelial cells are the ones that are lining the vessel, right? And they have these proteins called tight junction proteins, zonulin, occludin, the claudins, and they basically keep the endothelial cells zipped up, right? And there are things that erode those tight junction proteins, and one of them is inflammation. Now, you have the same kinds of barrier proteins in your lungs, in your gut, in your skin, in your nasal epithelium. And environmental exposures, inflammation, quite possibly pollution, all of them act to erode these proteins. Also, as you get older and you go through multiple infections and multiple exposures, right? What I call the dance card of life, you end up eroding these barrier sites. So what that happens is when you have breach of those, then the communication that was normally very tightly regulated becomes dysregulated. And so now you have substances that are leaking in that shouldn't be leaking in into the brain. As an example, gut inflammation makes those tight junctions erode in the gut. And what happens is the LPS, the endotoxin from the bacteria that normally live in your gut, leaks into your circulation. And now it's circulating in your blood, and that makes the BBB leakier. So anytime you have a leaky gut and signs of endotoxin circulating in your plasma, you know that there's something wrong, and that is going to erode your BBB. So that's how we think that chronic inflammatory conditions like diabetes, metabolic syndrome, obesity, how they are likely increasing risk for these age-related disorders like Parkinson's and Alzheimer's. The epidemiology, which is not to imply causation, but the epidemiology is really interesting. People who have had these autoimmune conditions, psoriasis, rheumatoid arthritis, Crohn's, IBD, and have been treated with blockbuster anti-TNF drugs that we were just talking about, right? Those drugs like Enbrel, Remicade, Humira. Those have been on the market for about 20 years, 25 years.
Okay.
If you look at the people who consume them because they had a prescription or whatever, and you look at the incidence of dementia or Parkinson's in those groups, the risk is about 50% lower than people who didn't take those drugs, right?
Right.
So what does that tell us? Well, it doesn't necessarily tell us that inflammation in those diseases causes Alzheimer's or Parkinson's, but it tells us that inflammation in your body in middle age can affect the risk of older onset brain diseases. And the weird thing is that these drugs have very little brain penetrance. So how are they benefiting the brain or reducing incidence of dementia if they can't even get in? So there's a couple of options. One is they don't have to, and it's really the peripheral inflammation. So if you are treating the peripheral immune cells and they're kind of quieted down and not inflamed, they're going to have a better conversation with the microglia in the brain. Second option is they do get into the brain because people with these conditions have leaky guts, like Crohn's, and they have leaky BBBs, right? Because of the inflammation in their bodies, now these drugs are going to penetrate in a way that they wouldn't in an uninflamed person. So it's a silver lining benefit from having these conditions and being treated for them early in life and then benefiting later. So I think that's fascinating. What it also tells me is that maybe these brain diseases did not start in the brain. Maybe they started in the periphery. Maybe you have a condition where you have metabolic dysregulation, you have other inflammatory conditions, and of course, you don't really pay much attention until you lose your memory, until you become disabled with tremors, right? So the way we're thinking about these diseases is this thinking outside the brain is what are the chances that organ dysfunction elsewhere in the gut, in the liver, in the lung, in your skin, based on exposures, pesticides and other chemicals, what are the chances that they are the contributors over many, many decades, because we've lived too long, right, to these brain diseases? So that is both daunting, but also exciting, because if these diseases start outside the brain, then the pharma companies that have left the CNS space because they can't get the drugs into the brain need to start thinking about how to treat outside the brain and how to treat it earlier, right? How to diagnose it earlier, which leads you right into prevention, right? Are we going to be able to prevent these diseases better than treat them? Absolutely. What neuroscientists didn't realize, maybe because we're too proud or have big egos, is that because we're brain scientists, right? That the same thing that benefits your heart, like a good diet, exercise, and fights cancer.
Sleep, good night's sleep.
Is the same thing that you need for brain health. If you don't sleep, the toxic aggregates aren't cleared by the lymphatics. If you don't sleep, you're not going to be able to, reboot the system, the garbage can, and flush the toilet. So it's important to have some humility and realize that it's not always about the brain and everything starting in the brain. So that kind of thinking, I think, leads you to come up with alternative therapies, right? If you think that the problem is the plaque buildup, then you're going to make drugs to remove the plaque, right? And one of the things we think about is, how did the plaque get there? Well, it turns out that based on genetics, some of the microglial genes suggest that maybe they're not fully functional from the time you are born, right? Maybe you're born with lazy microglia genes, and it doesn't matter if you live 30 or 40 years, but if you live 70 or 80 years, the accumulation of stuff that they haven't been removing from the household makes your brain like a hoarder's episode. And then you have to think about, okay, immunotherapy may remove the plaques, like take out the garbage. But if you don't fix what's causing the garbage to accumulate, it's going to accumulate again. So we think that the emphasis should be on the housekeepers, on the sentinels that are keeping things in a good working order and clean, so that you can reboot the vacuum cleaners, so you can rehab the parents, the glia, the immune cells that are taking care of the dependent children, which are the neurons, right? And
I love that analogy.
And doing that, you think, gosh, it's not always about the patient, right? The caretakers and the caregivers, there's like major exhaustion, and they're also getting older, just like your neurons are. So that has caused us to think about, how do we intervene earlier? How do we look for the immune system for clues that something is going on wrong in the brain? Because the neurons do send off signals to the surrounding microglia, but some of our work suggests that the ligand receptor expression in microglia and peripheral monocytes changes when you have inflammation in the periphery. That when you don't have inflammation, microglia are talking to each other mostly. But when you have peripheral inflammation, the microglia change, and now they're talking to the monocytes in the periphery. Why? Maybe they're saying, hey, what are you guys doing out there? It's going to get hot in here. You better quit it, or you better come help, or whatever. So we don't know enough about this crosstalk. We know that it's probably a normal process that's important for biology. But I think that we need to think about, well, what are the things that might lead to too much protein aggregation? What are the things that might lead to not enough trophic factor production? And we think that it's aging of the immune system, chronic infection, chronic exposures, again, the dance card of life. And to keep that healthy, you could be doing, things like, more exercise, generate exerkines, and generate more neurotrophic factors, and lower your inflammation, or making better choices with diet, and cutting down on the refined sugar, because it turns out that's the bad actor, not so much the fat, but the refined sugar.
Hey everyone, hope you enjoyed this episode of the No Brainer podcast, and see you at the next one.